noise during the exam. Actually, I hope it wasn't too disturbing. People saw I would. For this point, last few lectures in the course, we sort of finished our journey now through endoderm, mesoderm, and ectoderm. And so I wanted to talk a little bit about a couple of other topics that are related to developmental biology that also extend on into adulthood, including sex determination. So this is all the developmental biology that goes into our primary and secondary sexual characteristics, which we'll talk about. And then in the last 2-3 lectures, we're going to turn to plants. And now that we know quite a bit about animal developmental biology, some of the mechanisms, some of the paradigms, we'll switch over to plants and sort of do a compare and contrast about what's similar in that situation and what might be a little bit different. So hopefully this will kind of round out the course, and we are actually almost done. So it's really only going to be in the rest of this week and next week. So if you haven't had a chance, go ahead and look at the final from this spring, which is posted on RAMCT. And I think this is a good starting point for thinking about studying for the final. It's just going to give you an example of what the scope is and kind of the level of questions for the cumulative part of the exam. So not too early to have a look at that.
Well, it's next week, right?
Right.
It's a week from today.
It's a week from today, so definitely not too early. Okay, so today is going to be 2 different lectures on sex determination, and the first one is about sex determination in mammals. And in mammals, we have a situation where sex determination is strictly chromosomal. So by sex determination, this means what are the factors that are going to cause us to be male versus female. So what are the factors, the genes, the mechanisms that would establish these cells or these bodies as being the male phenotype versus the female phenotype? How do we do that? And in mammals, when we say it's strictly chromosomal, we mean that the mechanism for doing that relies on genes. So in other species, which we'll briefly touch on, sometimes you can have factors in the environment that are the key triggers for male versus female. But in mammals, it doesn't matter what the environment is, it is just strictly based on what sort of DNA you have present in your cells. So this is a situation, I'm sure we've known this since 5th grade. Females have X and Y chromosomes. Females have XX chromosomes. I'm going to go back to 5th grade. Males have X and Y chromosomes, and females then are the ones that make only an X gamete, whereas the males can make 2 different types of gametes, an X gamete or a Y gamete. And so it's actually the male that is responsible for the next generation determining whether that individual will be a male or female. So it's just strictly Mendelian inheritance here, and you get these 2 possibilities. So by contrast, other animals use different ways to accomplish the goal of establishing different sexes. And here are just some variety of situations that can exist. So in some insects, such as the locust, we have what's called the XO system. And in this scenario, there actually is not a Y chromosome. And what is different between the males versus females is simply that the females have 2 X's, where the males only have 1. So for every other chromosome, their males are diploid, but in the case of the X, they only have 1 of them. So 22 autosomes plus single X. Then in birds, we have what is actually the same situation as mammals, only the sexes are reversed. So it's the females that have gametes of 2 different kinds, a Z gamete and a W gamete, and it's the males that have 2 Z gametes. So there's no a priori or fundamental internal reason why males or females have to be the ones that make the same or different gametes. And so mammals have done it one way, birds have done it the other way, and it works the same in terms of passing on the genetics to the next generation. Then we can also have the haplodiplo system, And in this case, not only the X chromosome, but in fact the entire genome is either in the diploid state for females or in the haploid state for males. So, however, all three of these are relying on X chromosomes, Y chromosomes, Z chromosomes, W chromosomes. So these are all further examples of sex that is determined based on the chromosomes. So in the spring, I have another lecture that talks more about the effects of environment on development. And in that, I show some examples from alligators and amphibians where it's actually the temperature of the eggs when they're being incubated early in life that will affect whether they become male versus female. So that's an example of something that's not chromosomally determined for sex, but in fact it depends on temperature or an environmental factor to establish what that sex would be. And then if you think about things like global warming being pretty severe in certain areas, and these temperature differences between the two sexes are pretty small, only a couple of degrees, then that may eventually become problematic for some species to have the correct ratio of male versus female. Okay, back to mammals. We actually have two different stages to our final product here of being male or female. And stage one is called primary sex determination. And this would describe everything that is needed to make either an ovary or a testes. Okay, so it's going to reference back to genes. And in the fetus, they're building these two different types of organs, ovaries or testes, and this process is known as primary sex determination. However, just the existence of the gonads is not the only thing that marks us as male and female. And so secondary sex determination refers to everything else. And this is going to include some events that happen in the fetus, such as building the reproductive tracts. So, for example, the uterus, the vagina, all the female-specific types of structures, fallopian tubes, or in the case of the male, some of the glands and male reproductive structures, testes and so forth. These start in the fetus, and these are considered part of secondary sex determination. But then also, once we hit puberty, this process continues. So these are all processes that are mediated by endocrine signaling or hormonal type of signaling. So especially once you get into puberty, you have hormones that are stimulating some further growth and development of not just reproductive tract, but other parts of the body too. So think of stature, breast growth, beard growth, voice, all these kinds of things are part of our secondary sexual differentiation, driven by hormones. Okay, in the fetus, it's kind of interesting that at early stages, even though there is a difference in chromosomes, the anatomy initially looks the same. So this is what's called the indifferent gonad, or the bipotential gonad. So bipotential means that this gonad could go either way. It's going to either go towards the male phenotype or the female phenotype. Once those genes that are needed for making that decision start to get activated and the products get busy, or it could go to the female. So indifferent just reflects the fact that we can't tell one or the other. So what we see here is two structures called the metanephric kidney, shown here in gray. And this is something that's going to come from intermediate mesoderm. And that's another lecture we didn't get to do in the summer here, so we just have a shorter time. But the take home is that this intermediate mesoderm is going to give rise to kidney and to gonads. So we'll see these two types of structures next to each other in the fetus as things are developing. Okay. So, and then there's a set of two different glands. In green here, just for illustration purposes, is the Wolffian duct. And that's going to be right next there to these area where the gonads are growing. And then in orange or yellow is the July30 duct. And so initially, in both males and females, we have both sets of ducts are created and are present. Then, eventually, if it's going to be male, what we'll see is the Mullerian duct starts to degenerate. So that tissue just kind of falls apart and is resorbed into the body. But the Wolffian duct here in green will persist. And that will give rise to a portion of the male reproductive tract, specifically some of the tubules that are going to carry sperm from the testes through the rest of the male reproductive tract. So they're right next to developing testes, which is where they should be. And then in the female, it's the opposite situation. This time the Mullerian duct will persist and the Wolffian duct will degenerate. The Mullerian duct eventually goes on to form the oviduct of the female body system, and those are also going to be next to the gonads, which in this case are developing as ovaries. So now if we look at a cross-section through the embryo at about midway through the trunk, this will be the area where the gonads are developing. And so I've marked a circle up on top to show you where the notochord is. So if we're thinking in the embryo, we have our neural tube. Beneath that, we have the notochord. And then beneath that, we have the aorta, which is the major blood vessel. And then you can see the dorsal mesentery hanging down there, and below that would be the gut tube. So we have all those structures we've talked about so far. Then next to the midline is this region called the intermediate mesoderm. And you can see there the Wolffian duct in this case, which is going to be a male case. And then that little intermediate mesoderm, there's an area known as the genital ridge. So it's a specific portion of the intermediate mesoderm that would eventually give rise to either testes or ovary. So at 4 weeks, we're still in the indifferent gonad stage. So we just see basically cells within this genital ridge are proliferating. And as we get on to 6 weeks, still in the indifferent gonad stage, we start to form these sort of loose conglomerations of mesenchymal cells. So we have, these are called the primitive sex cords, and they're just the first proliferation of these mesenchymal cells, the first visible organization within these structures, which still applies to both sexes. Okay, beyond about week 7, we're going to start to see some differences between male versus female. So in the case of, let's see, this one is the female. No, this is a duplicate side. Keep on going. If the fetus is XY, so we're going to develop as a male. The primitive sex cords now start to align as tubules. So they're called the testes cords, and they form this whole interlinked network of tubules that become contiguous with this green Wolffian duct here. And so these are going to be the first structures within the testes where sperm will eventually be made. And these will be the seminiferous tubules down the line. And so in the fetus, they're already being established for the first time as these testes cords. So if you think back to that gametogenesis lecture where we talked about all the seminiferous tubules, and inside those we had the diploid cells that were going through meiosis to make spermatozoa. That will happen inside of these structures once we get to puberty. Okay. Meanwhile, the Mullerian duct will begin degenerating. Okay, if we have a female, so if we have a genotype of XX, we start out with our cortical sex cords, but instead of continuing to proliferate and forming tubules, these instead just take on more of just a loose mesenchymal configuration. And all of the black dots here, these indicate the germ cells, so the cells that are actually going to give rise to eggs. And then all the red cells are the surrounding cells that are going to become thecal cells or granulosa cells. So if you again think back to that lecture where we talked about in the ovary we have follicles that are developing, and inside of the follicle will be a primary oocyte, a secondary oocyte, and so forth. All of those other cells, the diploid mother cells, those will be granulosa cells or thecal cells. And these are already coming into place in the fetus. And we also talked about the egg cells themselves, how they start going through meiosis and then they arrest at a certain point. So that's kind of happening right here too. So, and then in this case, the Wolffian duct will generate mammillarian process. Okay, so now I'm going to talk a little bit about the germ cells. So these are the cells that we'll find in these gonads that are going to give rise to either sperm or egg. And the somewhat surprising, well, a couple of things actually that are different from mammals, different from the organisms we talked about. So when we talked about C. elegans, we talked about the cells that had inherited the P granules. Those were going to become the germline of the embryo. So they had this morphogenetic determinant really, really early in embryonic life, already at 4-cell stage, already at 1-cell stage, and that was setting aside a certain group of cells that then eventually became the germ cells. Same thing with Drosophila. They formed the full cells, and those eventually became the germ cells. But in mammals, it's a little bit different. So there's not an early population of cells that are specifically sequestered to form the germ cells, but these will arise from the embryonic portion of the embryo called the epiblast. And so down the line, we'll eventually be able to identify a population of precursors that will form the germ cells, but it just takes a little bit longer for these to arise in the mammalian embryo compared to some of the invertebrate ones. So the other surprising thing is that when the germ cells arise, they're not actually in the gonads. They have to migrate to get to the gonads. So they're going to be initially specified at one point in the embryo, and then they're going to have to migrate through the endoderm, so basically through the gut area, and then when we get up to this region of the trunk where the genital ridges are located, then they'll exit out of the endoderm and go into the genital ridges and become colonizing there as the future germ cells. So in this picture here, we have these red dots that are indicating the distance that they have to go. So they're going to start out fairly posterior in the embryo and migrate all the way along the midgut area until they get about mid-trunk, and then they'll exit and go to either side into these two genital ridges. People have also taken advantage of GFP labeling to express GFP using a promoter for some gene that's expressed in germ cells. And so you can actually get to these germ cells and see how they're migrating through the embryo and going out and colonizing the gonads. And they seem to migrate just as these individual cells, a group of them going more or less together, but still, as individual cells, and they eventually get to the gonad. So I think that's kind of interesting and surprising. Okay, so we said that in mammals, we are relying strictly on genes to form the male versus female phenotype. And so what are those genes? And naturally, we have noticed that the inheritance of the Y seems to correlate with the male phenotype. But do you actually need the entire Y, or is there just a portion of the Y? And there are these rare people out there that have bits of chromosomes that have switched. So a portion of the Y might have swapped with an autosome, and so they don't get the full Y, they only get a little bit of it. And it was found that as long as you have about a 35,000 base pair region on the short arm of the Y, that's all that you needed to make a perfectly normal male phenotype. So this suggests that there's some critical gene or genes in this 35,000 base pair region on the short arm of the Y chromosome that is the critical bit. If you get these genes, then you're going to have everything that you need to go ahead and complete male development. So, 35,000 is still quite of a big area, so there could be multiple genes in there, and at first people had no idea what this gene or genes might be. And so they coined the term Testes determining factor to try to describe the critical gene that you would need to form the male phenotype, or to form testes, basically. So there's great interest in figuring out what this gene might actually be. And one good candidate is a gene called SRY, and this is found on the Y chromosome. And we now consider that this is in fact the critical gene, that it is the testes determining factor. And what it stands for is the sex determining region of the Y chromosome. So not really the most catchy gene name ever, but on the other hand, descriptive for what it does. And so this is a transcription factor. We see that it contains an HMG box, which is a conserved region that SOX family genes have. So this came up when we talked about endoderm, that there is a family of genes that have this so-called HMG box because they're a high mobility group and the proteins happen to migrate rapidly on gels. So this SRY gene is going to form a protein that will bind to DNA, and this particular gene family seems to actually bend the DNA as it's binding, and that may be just part of the regulatory process for the target genes and whether or not they're going to be transcribed or repressed. So this kind of makes sense that you would have a transcription factor as being the gene product that would drive, the whole process of organ development. We've seen some examples of that kind of thing before with the PAX6 gene driving eye development and the PDX1 gene driving pancreas development. So these are both transcription factors, and same thing here, SRY acting as a transcription factor. So what is the actual evidence that the SRY gene would be this critical testes determining factor? And to answer this, we're going to employ again our show it, move it, and block it experiments to gather evidence that would test this hypothesis. So first of all, showing it. So SRY gene is going to be expressed in males, but not in females. So that's a unique difference that we can just observe between males versus females. Males make it, females do not. What if we took the SRY wild-type gene and we inserted it on a transgene into an XX individual? So now everything about this is going to be female because they don't have a Y chromosome, they have two X's, except the only thing we're introducing now is the SRY gene, which will be also expressed. And so that's a movement experiment, actually. We've moved the SRY gene over into the female genotype and allowed it to be expressed. And this is what happens. So these are two mice, and they're hanging on a bar here just so we can look at their reproductive phenotype. And this is what male and mouse private parts look like. And here we have for XXSRY, mouse has exactly the same phenotype as the male anatomy. So these are, in fact, developing as male mice, even though genetically they are XX with the transgene. Okay? So what about other genes that might be involved in this pathway, possibly as targets of the SRY gene down the line? And one critical gene also happens to be in the same family. It also has this HMG box, and this is the SOX9 gene. This gene also, if you introduce it on a transgene, has the ability to introduce testes into XX animals. So let's look at what this looks like. We have here some organs taken from the body of these mice. K is the kidney, B is the bladder. And so between these two structures, you're going to have either testes, T is testes, or O for ovaries developing. This is, what we'll see is that in wild-type animals, we get expression of SOX9 within the genital ridge, and we get testes that are developing. In the SOX9 XY transgenic, now we see expression in the genital ridge, and again we get testes developing. Whereas in C here, this is the female phenotype, so no SOX9 expression in the genital ridge and ovaries, But then D is the critical experiment here, where we take the XX female genotype, and we're now inappropriately expressing SOX9 in the genital ridge, and then this is going to develop into testes. So that also has its ability to induce testes formation, even though it's an XX individual. So when this was discovered, people were saying, hey, what's the deal? Maybe we have two testes determining factors. But that is not going to be possible because SOX9 is on an autosome. And we already know that there's no correlation between which version of the autosomal chromosomes are inherited. Either mom's version or dad's version is different in every individual, and there's no correlation of this with whether you become male or female. There is a correlation between whether you inherit the Y chromosome and become male or female. So this could not be the testes determining factor, but it might still be in the pathway of the sex determining factor. So probably a target at some level, direct or indirect, of the SRY gene. So this is the model here, that from the Y chromosome, we're going to be in males expressing the SOX9 gene. And so here's the open reading frame, including the HMG box. And when this thing is made into a protein, it would bind to specific sites in enhancers or promoters of the target genes. And one of these target genes might be SOX9. And so then SOX9 protein would get made and go ahead to bind even further targets down the line. So actually SRY binding to SOX9 and SOX9 protein then binding to further target genes. Okay. Yes?
What about an XY double knockout mouse?
Okay, so if we had an XY mouse and it had this SRY gene knocked out,
Yeah.
so that would be lacking the SRY gene, and that would develop as females.
Okay.
Although what we're going to see, we'll kind of come back to that, is that you actually need to still have active gene-directed processes in order to get a fertile, fully naturally formed female. So it's not just the absence of SRY, but also some other factors
So
as well.
it would be an infertile female?
It can be an infertile female, yeah. Okay, so I'm going to show you a model here in a minute that kind of gets to some of the genes that are involved. And one of the genes that it will talk about is a gene called R-spondin. So I just wanted to first introduce what this is. So this is the Wnt pathway that we've seen before, where Wnt paracofactor binds to its frizzled receptor, and then we either have beta-catenin being degraded if Wnt is not present, or if Wnt is present, then beta-catenin can go to the nucleus and interact with target genes, and you get differential gene expression as the result of this intracellular signaling information. So the R-spondin, this is obviously a second paracofactor. It has a receptor, and what it seems to do is actually potentiate or sort of synergize or upregulate the Wnt pathway. So when these two are both active together, you get sort of a much stronger, much more vigorous production of beta-catenin. So R-spondin is something that's going to synergize the Wnt signaling pathway and make it go smoother or better. Okay, so here's our model that is the current vision of how sex determination works using all the things that we've talked about so far. So we have two situations. Blue is the pathway that would lead to testes development, or pink is the pathway that would lead to ovary development. And initially this is going to start out purple, it's not really pink, not really blue, it's something in between for both genotypes. Then the key situation will be whether the SRY protein is made or not. And of course in males it will be made, and then that stimulates the production of SOX9. And at this point we get to a model that I call the make A but don't make B. So the make A means we're making a positive proactive choice to keep certain genes turned on. So SOX9 is going to stimulate FGF9, and these two get going in a positive feedback loop that keeps both genes expressed and male development chugging along happily. Okay, SOX9 is going to also hit some other downstream targets that are proteins needed for male development, other testes-forming genes. They'll also start making an anti-Mullerian duct hormone so that the Mullerian duct can get degenerated, because we will want to get rid of that one and keep the Wolffian duct in males. So these are all positive, proactive, make situation A type of scenarios. On the other hand, don't make B means that it is inhibiting beta-catenin production within these cells. So that tends to shut down any possibility of the female morphology getting started at the same time. So the cells are making a choice here. We're going down testes or we're going down ovary. And you need to actively pursue one, but also actively shut down the other choice, seems to be. Okay, so if this were a female, now there's not going to be any SRY, and there's not going to be any SOX9, and so there's no repression of beta-catenin. So beta-catenin gets going gangbusters and starts doing proactive things like forming other ovary genes, turning on some female-specific targets, and specifically making this R-respondent gene, which will get the Wnt pathway up and running, which is an important part of female development. So they also are going to then feed back and repress male development. So the same thing, like they need to actively pursue the creation of the female morphology through the expression of appropriate genes, but then repress the possibility that we're going to get male development. So there's a lot of crosstalk between these two options to make sure that one variety stays on and the other stays off. Okay, so all of that business is really part and parcel of the primary sex determination, because we said that's the decision of making either an ovary structure or a testes structure. But meanwhile, there's all this secondary sex determination that's also going to begin in the fetus and then ultimately carry on in the young adult. So the formation